Good afternoon. My name is Eliram Reyes-Powell,
And I'm Carmen Puentes.
and we are going to be presenting our remote internship at the University of California, respecting protein modeling and molecular dynamics, and we're from Montreal High School.
So there's only a very small amount of proteins that have been discovered or created through evolution, and the possibilities are astronomical compared to the different amounts that can be created. So by studying proteins, many pharmaceuticals can be created. The medical field would completely change. The material industry can be completely improved. Yeah.
Okay, so the overall impact of this internship was learning about how to develop cardiac arrhythmia drugs, and the main ion channel of study was the hERG ion channel for potassium, which is the one that causes cardiac arrhythmia. And we learned about the protocols and about just how it is that we could develop a potential drug candidate for cardiac arrhythmia. Okay, so our 6 enlightening weeks started at Yarov's lab. We worked with Professor Yarov for 3 weeks, and we started by playing a video game called Fold It, which explains to you the protein interactions, and you can just learn the very basics of how protein modeling works. And we proceeded with Chimera and Barbera, and with Chimera, we were able to just see the environment in which a protein could be edited, and we also learned how to clean a protein to then be used for the Barbera software, which Barbera is found in the secure shell from UC Davis, and from there we were able to use the Rosetta software to edit proteins and relax them and run the JE ligand dock protocol. So for Igor's lab, we started by studying molecular dynamics, and that was through the visual molecular dynamics application called VMD. And for that one, we learned basic protocols, and we learned a basic software that we could use to just simulate how a protein would denature, how a protein would interact with an environment over a period of time. And from there, we started to study a protein called valinomycin, which was back then being studied for potential COVID-19 drug development or treatment development. And we ended that part of the internship by using a software called CharmGUI, which allows you to create the environment for your protein and allows you to just create a lipid bilayer and create a water environment.
And our last week was at Colleen's lab, in which we actually went in and studied the deep layer of the cell wall. We looked at the physics implications of the cells and the ion channels, and we used electric circuits to model the cell membrane and study the specific ion channel that Eliram mentioned before, called hERG. And we just used ventricular potential action potentials, and we created a program through the use of C++.
And some surprising events. During our internship, we had the RosettaCon 2020, which was a conference in which we were able to get to know about the latest developments in the protein modeling field, and we were able to connect with the experts who are in the frontiers of this field. And we also received an invitation for continued work, which is an opportunity we have for life.
So our advice for future interns is to not hesitate about applying. You never know what's going to happen. If you're very interested about the STEM field and you're passionate about it, you should always apply. Be prepared to be challenged. This is a very challenging environment, and you have to be able to ask questions and be mentally prepared for it. And also prepare beforehand, study, and make sure that you have the basis of what you're going to be learning for the next 6 weeks. Thank you.
Thank you.